All right. Welcome, everyone. I'm excited. Welcome to the CCMB seminar. We have another one next week, so come to that as well. Okay, so I'm very pleased to introduce today's speakers, Dr. Molly Schumer, who's a professor at Stanford University. Molly earned her PhD at Princeton in the Department of in Evolutionary Biology, right? At Princeton, and was co-advised by Peter Andolfato and Bill Rosenthal. And then she did a postdoc with Molly Przorsky before joining the Harvard Society of Fellows and working with David Bright at the medical school as well. And then as postdoc, she was named an HHMI Hanna Gray Fellow. Molly's lab uses natural swordtail fish populations as a model system to uncover the genetic mechanisms that drive adaptation and speciation. Her group integrates genomic, molecular biology, behavioral, and physiological techniques to understand how new species form and how they adapt to their environments. And Molly has made several very impactful contributions. And as recognition, she has received many awards that are too many to mention them all here, honors and fellowships. And her work is funded by HHMI, NSF, and NIH, as well as private foundations. And so please join me in welcoming Molly Schumer.
Can everybody hear me? Okay, great. Yeah, so thanks for the introduction, Amelia, and inviting me. Yeah, I'm excited to tell you all about my lab's work. I apologize in advance to everybody going to the supergroup retreat because this will be a duplicate talk, but hopefully hopefully it'll still be interesting. Okay, so before I launch into my talk, I want to acknowledge everybody in the lab, past and present, who have been pursuing a big diversity of questions, kind of ranging from genome evolution to trait evolution, and a little bit into behavior and ecology. And I also want to thank the team of staff scientists in the lab who have been really integral in sort of trying out new and kind of high-risk things, and many of them who have gone on to grad school over the years. So I'm going to focus on a subset of projects today, led by grad student Tris Dodge, who's hidden by my icon, Nadia Hagani, Will Pangburn, and postdoc Paola Fascinato-Zago. But wanted to start by acknowledging everybody and what a fantastic team they've been to work with. And in the projects I'll talk about today, I'll also be touching on the work of many collaborators and their labs without whom this work wouldn't have been possible. Okay, so as Amelia mentioned, my lab is really motivated to understand evolutionary processes at the molecular level. And we're really fascinated by, like many evolutionary biologists, by this variation we see across the tree of life, how it's maintained in distinct species. And our work investigating this process of diversification focuses on two major areas. So a central question in our research and in evolutionary biology more generally is how organisms adapt to their environments, the environments in which they're found, and then how over evolutionary timescales do they diversify into new species and maintain that diversity to populate the tree of life. And one sort of kind of gap that I think our lab has been working to fill is that sort of historically, there's been kind of a separation between fields and approaches in really understanding the connection between molecular mechanisms and evolutionary processes. So, and there's good reason for this. I think that researchers who relied on lab models have been able to make a lot of progress on understanding exact molecular mechanisms underlying different processes and traits, but then haven't necessarily had access to the natural populations where those processes are unfolding. And on the flip side, people who have been able to study things in an ecological and evolutionary context sometimes lack the tools to study these processes mechanistically. And so one of the goals of our lab group has been trying to merge these disciplines with a high power system. So I've been trained in both molecular and evolutionary biology as a student and postdoc, and really want to bridge this gap between what's underlying these processes and how evolution is actually occurring in nature. Because I think that the genetic mechanisms through which evolution is acting are, have these ability to constrain the evolutionary process, and in turn then influence the molecular mechanisms that we're studying in the lab. Okay, so to answer these questions, my lab has been really focused on developing this new model system that I'll tell you a lot about, where we can do both things, where we can really build powerful lab models to do experiments and study evolution and mechanism, but also go back to the natural populations and ask how these processes are unfolding in practice. And so I'd like to introduce you to our model system, swordtail fish, or Xiphophorus. This is a model system that we've been developing from the ground up, from collecting fish to sequencing genomes, to developing sort of long-term monitored field populations. So swordtails are this very diverse group of fish found mainly in Mexico, a little further south into Central America. They have this amazing morphological diversity, but they're also a really great system to study how new species form and persist, and a great system in which to study adaptation to the environment. Okay, so I want to tell you a little bit sort of about the background of the system before diving into projects. One of the reasons I started working on this system as a graduate student is they were one of two freshwater fish species that had a sequenced genome at the time. And that really enabled us to start developing the genomic resources. And as a graduate student coming in from more of a genetics and molecular biology background, I got thrown into fieldwork and all the excitement of like muddy boots ecology as well. Since starting the lab, we've been really working on kind of transforming this into a model system. So I think they're a perfect system to bridge this gap that I mentioned. They're small fish, about the size of zebrafish. We have over 5,000 of them in lab of 12 different species. And so that gives us a lot of power to look at these different phenotypes. Their genomes are small. They're about a quarter of human or mouse. They have short generation times. So we can cross them, do experiments, manipulate them in the lab. And then one of the major things we've been working on in the last several years is developing molecular tools to be able to do gene editing and other types of manipulations. And then crucially, we can study their natural populations and ask how evolution's unfolding in practice. Okay, so today, for the purposes of time, I'm really going to focus on this question of how organisms are adapting to their environment, what molecular mechanisms are underlying these changes, and how they evolve. And the set of traits that I'm going to focus on today are coloration or pigmentation traits. And these traits can be very important for both survival and reproduction. They are important in crypsis. They can be important in the ability to find mates and have social interactions with other individuals. And there have been many studies on the impacts of pigmentation in natural populations, and some, like studies of deer mice, have really been able to link that to genetic variation. And I think this is an excellent system to study these kinds of questions because of thinking about the genetic architecture of pigmentation. So pigmentation traits have an advantage from a technical perspective because they tend to have a simpler genetic basis than many of the traits that we study as evolutionary biologists. And this might be because of the structure of the developmental and genetic pathways that lead to the differentiation and maintenance of different pigmentation cells. And I would argue that this is actually a really helpful property when you're interested in studying evolutionary history, because you're not trying to necessarily track hundreds of loci spread throughout the genome and understand how they evolve. You can have sort of a more modular genetic architecture that you're then able to go deeper into understanding the evolutionary origin and consequences of the trait. Okay, showing this slide I showed a few minutes ago just to highlight that swordtails themselves are a really excellent system for studying variation in pigmentation. There's massive variation within and between species in a variety of visual phenotypes. Most of these that you can see here on the screen are under sexual selection, but some can be very important in the ecological environment as well, and certainly quite a few of them are important in social interactions. So the first project that I'm going to tell you about started quite unexpectedly. So we were in Veracruz, Mexico, where some of our field sites are, and we were collecting from these different rivers. We were looking for more diverse population samples of a particular species that we work on in the lab. And we were fishing near the town of Juntas Chicas, which is a very small town, and started pulling up fish that looked very unique. So Nadia Hagani, who led this project, basically was sending me pictures from the field of fish that look quite normal for a swordtail and fish that looked like this, with these sort of stripes of reflective scales along their bodies. What we were able to document collecting over multiple sites is there's quite a lot of variation in this pattern, but there's very common phenotype in this region of these really bright iridescent scales. And Nadia quickly named this the sparkle phenotype. After some arguments with lab members, it stuck as our name for this phenotype. So I'll refer to this as the sparkle trait throughout the rest of the talk. Okay, so this gives you a sense of the phenotypic variation at the organismal level. Nadia actually comes to our lab through the cellular and molecular biology program. And so she was really interested in kind of diving deep and characterizing this at a cellular level. So she dissected scales off of iridescent and non-iridescent individuals. And what you can see is in these iridescent individuals on the scale, there are these sort of mass of cells that is absent in the non-iridescent individuals. And zooming in on these, based on their morphology, they look a lot like a cell type called iridophores that have been described in other species. And so iridophores are a cell type that basically generate iridescence by reflecting light using subcellular crystals. And based on her images, it looked like that was likely what was going on, but she collaborated with a colleague to do transmission electron microscopy so we could actually image inside the cells and see if those crystals were there. And so this is a really beautiful image of an iridophore cell from these scales, where you can see these little kind of crystal structures inside the cell. And in other species, those tend to be guanine crystals. We don't know what they are in our species, but these are actually interacting with light and generating this sparkle phenotype that we see. Okay, so that was very exciting. I never thought we would do transmission electron microscopy in my lab, but kind of gave us a really clear idea at the cellular level how this trait is being produced. She also tracked this trait developmentally. And one thing that was quite interesting to us, perhaps less so to people that don't work on these fish all the time, is that in Xiphophorus in general, many pigmentation traits show up around sexual maturity. They're under sexual selection. They're really important in mating interactions. This is a trait that shows up much earlier. So we see sort of the first signs of it in fish that are about a month old, sometimes as young as 2 weeks. And it shows up as these sort of migrating cells that kind of burst through the surface of the skin here. And over time, they kind of fill in until you have the whole scale being iridescent. She tracked a bunch of individuals, about 50 over time, to show that there's a really predictable pattern in where this trait first emerges along the lateral line. And some individuals get this sort of secondary stripe or some pigment in other spots of the body, but this tends to be that area where it first emerges. Okay, so this gives us a sense of what the phenotype is, how it's emerging developmentally. Now that we understand how this trait is sort of generated on a cellular level, we're interested in transitioning to understanding its genetic basis and how it arose from an evolutionary perspective. So Nadia collected about 300 fish, and we did a genome-wide association study and see this really clear association on chromosome 15 that explains basically all of the variation in the trait presence or absence. And so this, again, sort of going back to this pigmentation trait idea, gives us a really clear place where to look in terms of understanding the genetic changes in evolutionary history of this trait. Okay, so in this associated region, there are about a dozen genes. And as we began just sort of looking through the annotations, we were really excited about a particular one called ALKL2. And this is a gene that's known to play an important role in the differentiation of these what are called pluripotent progenitor cells, but basically the cells that lead to these different pigment cell types into iridophores as opposed to other pigment cell types. And so that seemed like a really exciting candidate. It's also a gene that's not just important for the differentiation, but also the maintenance of the trait. And so what Nadia did next was sample scales from sparkle and non-sparkle individuals and do bulk RNA-seq on several replicates, and then look at patterns of expression around our genome-wide association peak. And so what she sees is very nice and clear, where we have in the iridescent scales strong upregulation of the ALKL2 gene compared to individuals without the iridescence. And we see no difference in expression in any other gene in our genome-wide associated region. Genome-wide, there are about 70 differentially expressed genes. Most of them are iridophore related. Our hypothesis is that this is probably the driver. That's what's driving our genome-wide association peak and then regulating these sort of downstream effects. One thing that we were unsure of when we were looking at this result is sort of what what was going on throughout development, because we did this RNA-seq project in adult individuals, and so Nadia used qPCR to basically look at a bunch of individuals throughout development, and what she did actually was genotype them to make sure that they had the right genotype to ultimately develop sparkle. And what she sees in the babies and juveniles, which don't yet have the trait, is that there seems to be some sort of pre-patterning. So even though they don't have the trait yet, they are upregulating this ALKL2 gene long before the trait itself develops. And so, yeah, so we think it's sort of laying the groundwork in terms of expression for ultimately developing the trait. Another approach that Nadia took, which was very cool and new to me, was to use pharmacological inhibitors to sort of disrupt the pathway in adult scales. So she basically designed an approach where we could culture adult scales and keep the cells and iridophores alive in vitro. And then in a subset of scales, she added an ALK inhibitor, so an inhibitor of this pathway. And what she sees is very quickly, within about 12 hours, the iridophore cells die and kind of collapse into these punctae. And she has some really cool time-lapse videos of this happening. And so that's sort of another piece of evidence that this pathway is important for maintaining this phenotype. In scales that weren't treated, we can keep them alive for several days in culture, but the treated scales have this really rapid cell death post introduction of the inhibitor. Okay, so hopefully I've convinced you now that we think ALKL2 is this gene that's driving this difference between the sparkle and non-sparkle individuals. And so the next question for me is how did it come to regulate this trait? What's the evolutionary path that it took? And so, we thought it was likely that there's some kind of cis-regulatory difference driving this pattern, because we see the GWAS peak and then we see differential expression of a gene under the GWAS peak. And in order to look at this in more detail, we generated haplotype phased assemblies using long-read data for 3 individuals with the trait and 3 individuals without it. And I'm going to show you on the next slide sort of a cartoon of the individuals we sequence. So these are paired by individual, so it shows both haplotypes on chromosome 15 for that individual. And on the top are the individuals with the sparkle trait, the bottom are the individuals without it. And so what we observed is that all of the individuals we sequenced that had the sparkle trait have this large 18 kb insertion in at least one haplotype. And we subsequently used PCR of the insertion breakpoints to confirm that the haplotype, that this insertion is consistent across dozens of individuals with the sparkle trait and absent across individuals without it. And so, of course, our immediate question is, what is this insertion? After blasting it, we knew immediately that we needed help because it blasted to a transposable element family. And I knew we were out of our depth immediately, so we reached out to Guanju Han, who's done a lot of phylogenetic analysis of these types of transposable elements. Basically, Guanju was able to do phylogenetic reconstruction of this transposable element sequence with many other known classes of transposable elements, and was able to classify it as an endogenous retrovirus of the foamy class. And so these, again, I felt very out of my depth. I didn't even know such things existed. These are viruses that infect an organism and then can insert in the genome and basically become transposable elements. So they're acquired through an infection initially, but then they vertically transmit and spread throughout the genome. That was really exciting. It's actually started like a whole new branch of research in our lab, investigating the function of these elements in other parts of the genome. But getting back to this question of how this might be regulating expression, I'm showing you the alignment in a slightly different visualization here. You can see this 18 kb insertion. And then on the x-axis are the genes that are present in this region near the insertion. And we can see ALK-L2, this gene I've been talking about, important in iridophore development and differentiation. And then the insertion is actually not in that gene, but in the next gene over, ACP1. And what's interesting is it's inserted in the antisense direction. And this actually turns out to be very important because there's often strong selection for these intronic insertions not to be in the sense direction because you'll disrupt the protein product. But it is in the same orientation as ALK-L2. And so this raised the possibility to us that it might be acting as an enhancer of ALK-L2. Note that we never see that ACP1 is differentially expressed in our RNA-seq data, nor in qPCR that we've done through development. Okay, so how would this insertion be acting as an enhancer? Well, again, I knew none of this when we started this project, but I've now learned that it's actually very common for these endogenous retroviruses to be co-opted as enhancers in the host genome. Usually this happens over deeper evolutionary timescales. For example, in humans, there are many endogenous retroviruses that serve as enhancers, but they're chopped up old versions of an ancient burst of infection. And so what we initially saw was that we have this typical structure of endogenous retrovirus where we have these long terminal repeats on either end, and that's important for the insertion of the virus, and then actually all of the coding elements that are required to generate an active element. And we focus first on these LTR regions, because these are the regions that are thought to be really important in enhancing gene expression, not just for the transposable element to replicate, but for the host genome more generally. And so we used an attack-seek approach to ask whether we saw patterns of chromatin accessibility that localized with these LTRs. And so I'm showing you on the top a non-sparkle individual, and on the bottom a sparkle individual. And what you can see is that there's this enhanced chromatin accessibility that aligns with these LTR elements. What was striking to us was this was actually the only difference between our sparkle and non-sparkle individuals across this region in chromatin accessibility. So we didn't see differences at the promoter of alkyl-2 within the sequence of alkyl-2, just within this LTR element of the retrovirus. Okay, this we thought was quite suggestive, but we decided to kind of push it a little bit further, and we did an in vitro luciferase assay where we took the LTR sequence as well as several other sequences throughout the region, and basically see that the LTR sequence is the only sequence that drives high luciferase activity in culture. And so this suggests to us that it's this sort of chunk of sequence associated with the transposable element that seems to be activating and maybe driving the upregulation of alkyl-2. Okay, so I already alluded to this, but another inference that was really exciting to us was not only are these LTRs important in regulating expression, but unlike most other cases where LTRs are playing a role in expression, this transposable element appears to be active. So I already mentioned it has all of sort of the essential proteins to copy itself and insert in the genome. We scanned these for stop codons, but we had sort of other routes to look at activity. So we saw that this was expressed in both the germline and the soma based on RNA-seq data. But even more compelling was sort of reanalysis of these long-read assemblies we did. So I've already showed you that individuals with the sparkle phenotype have this insertion on chromosome 15. But if you look across the rest of the genome, there are lots of insertions of this element. And most of them, with the exception of this one on chromosome 12, are polymorphic in our sample. And this is a really clear signal of an actively replicating transposable element, because if you allow sort of the evolutionary clock to run for long enough, these will be fixed or lost if they're not actively inserting. And so this suggested that this element is either active right now or recently active. And so we wanted to take advantage of this data set we had of many of these sequences throughout the genome to ask, what kind of evolutionary time period is consistent with these insertion profiles. And so we used the population genetic simulator SLIM, which many of you have used or are familiar with, to ask how old. Do you have a question?
Oh, yeah, sorry.
Go ahead.
Yeah, sorry. So I was just wondering, why are the transposable elements so regularly inserted across different samples?
You mean like there are so many on chromosome 5?
Yeah, and across all samples, to be fair.
So
the transposon hits similarly across.
So my interpretation is that there's a single insertion event, and then genetic drift or selection will modulate the frequency. Is that what?
Yeah.
Okay, cool. Yeah, so how based on the sequence differences between these elements, we can sort of, and the demographic history of these species, we can infer sort of a rough time of insertion. So one of the things that we see is these elements are extremely similar to each other, again, consistent with a very recent invasion. There are only, I think, like 6 substitutions found between any copy. And this is an 18 kb region, so that points again to a very recent invasion event. When we do sort of a grid search in SLIM, looking for trying to fit both the frequency and the divergence axis, we estimate about 15,000 generations since these elements started inserting and diverging from each other. And so this sort of, again, kind of hits home that these elements are very young, and they're kind of actively in the process of invading this genome. So at this point, know a lot about what genetic changes, what cellular changes underlie these sparkly individuals. But one of the things we're really excited about is kind of understanding how it's operating in the environment. And I would say, even though that's probably the motivating factor for this kind of work, it's actually the area that we still understand the least. But I think it's something that's very exciting because we can tell from observing these individuals in the lab and in natural populations how tunable this signal is. So this is the same fish, just angling its body in a different direction. You can see in one, it's very cryptic, and in another orientation, it's very conspicuous. And so we think that these tunable signals could be a really exciting way to understand the interaction between an organism and its environment. And so we've been working with colleagues in Puebla in Mexico who specialize on iridescence and trying to understand and characterize this signal better. And so one of the things that they were able to do was quantify the angle of light where the signal is brightest. And we see that a 90-degree angle sort of maximizes the brightness of an individual. That this is actually true across wavelengths of visible light. So we were quite interested in this 90-degree angle because it's suggestive of a signal that might be tuned to avian predators. A signal tuned to other fish would not be maximized at the 90-degree angle. Yes.
Like a standard position?
Yes, a fish like this. Yeah. But yes, this is another variable that we haven't, because they do move their body. And what, yeah, I don't want to go on too much of a tangent, but we don't think they know that they're iridescent, and which is too bad for them. And so they don't seem to have behavioral differences, which I'll come to in a minute, that distinguish them from the non-iridescent fish, but the signal they're producing is different. So the association with avian predators is really interesting because these are the major predators of swordtails in the wild. Okay, so to get directly to your question, actually, one of the things that Nadia has been working on, which again, getting into things I never thought I would be talking about, she built a model bird of a kingfisher, which is one of the most common predators of swordtails. And she hung up a string and she has it swooping over the tank. So if you let it go, it swoops over the tank. And the fish sees it and reacts to it. And the fish all react in the same way. And this is a very deeply conserved reaction in fish. So it's called a sea start. So once they're startled, they turn their body and they move fast in the other direction. It's a reflex. And so this behavior does not differ between individuals, but she's been able to capture on camera that when they do this, the sparkle fish often create this sort of flash of light. And so the question we're really interested in, but haven't really nailed yet, is, how is the predator going to perceive that? When they create this flash, does it look like light on the water? Does it make it difficult to localize the fish? And we haven't thought of a way to test this without really extensive IACUC protocols involving birds, but I'm excited to hear any suggestions you might have. So this is mainly to say this sort of work is in progress, trying to understand how this signal is interfacing with the environment. But we think there's a lot of rich ecological questions to pursue here. Okay, so I've just told you about this case where trait variation is controlled by this super recent transposable element insertion. And of course, this is only one of many ways we might expect trait variation to be controlled. So I'm going to switch to examining another case where very different evolutionary mechanisms are at play to maintain trait variation. And so returning to these images that I showed earlier of swordtails, one other sort of piece in thinking about the variation we see that's really motivated us is the distribution of traits. So a common observation is that we see sort of non-phylogenetic trait distribution. So very distantly related species that share a particular trait. For example, these species are called swordtails because many of them have this long fin. But these two species, Variatus and birchmai, have close relatives with these long fins. They're about 6 million generations diverge from one another, and they sort of have converged on this loss trait. Now, for a loss, that's kind of easy to understand. There seem like there are many ways to lose a trait. But we do see this pattern throughout the group of this sort of recurrent appearance of traits among distantly related species. And this, to me, is a very intriguing observation because the other focus of my work, which I'm not touching on today, is that these species frequently hybridize. And so this potentially, presents a mechanism for all this mixing and matching. Okay, so I'm going to get to the evolutionary mechanisms in telling you this story, but I'm going to start somewhere completely different. So hang on for the ride, and hopefully it'll be worth it. So we're starting first with reproductive biology of swordtails. So one of the traits that we observe this in is a trait called the false gravid spot. So this is a black spot that occurs in males on the abdomen. It's found across a huge range of species. And in all species in which it's found, it's polymorphic. So some males have it, some males don't. I'll first tell you a little bit about the genetic and developmental basis in birchmai, which is a species that has the trait, and then kind of return to the rest of the species and the evolutionary piece. Okay, so the reason we call this trait the false gravid spot is that it appears to be a mimic of a trait we see in females, which is called the gravid spot. So swordtails have a really unique reproductive strategy for fish. They have internal fertilization and live birth. And so this spot in females is actually caused by growing embryos, stretching a tissue called the peritoneum. And the reason it looks black is because you're basically kind of seeing through the female body and you're seeing the embryos. Now, obviously, males are not going to have this process unfolding, but what we see in juvenile males as they start to get toward sexual maturity is many of them will grow a similar looking trait. Now, interestingly, over time, these males will go on to develop other male secondary sexual characteristics, fins, colors that make it very clear that they're males, but they never lose this spot. As I mentioned, there are, in all species where the trait is found, there are other males that do not develop the spot, and they never will. Okay, so we know that in males, the spot is not going to be caused by developing embryos. How is it regulated? Well, we can see based on histology where the melanophores are accumulating, and that's what's generating the darkness that we see as the spot. But what was really interesting is sort of this particular tissue is a muscle and connective tissue that actually moves this fin. And this fin is called the gonopodium, and it is a fin that is used for internal fertilization. So the males release sperm packets onto this fin, and they use the fin to deliver those sperm to the female. And that's how fertilization occurs. And so what we have going on here is we have a male-specific tissue. So this tissue does not exist in females, that it connects to this fertilization organ. And this male-specific trait results in these males transiently looking like females. Okay, so we were really interested in studying this life stage where these juvenile males kind of resemble females before they develop other traits. and particularly interested in understanding social interactions between males with and without this trait. One of the major challenges we have in swordtails is male-male competition and mortality. So as males get older, they become dominant and aggressive, and we have a lot of problems with basically keeping males alive and not killing each other in our fishery. And so what we did is basically set up this experiment where we had a dominant male and two size-matched males, one with the spot, one without, and looked at the impact of the spot on aggression. And what we see is really clear. Individuals without the spot receive a lot more aggression from the dominant males than individuals with the spot. But what was quite surprising to us was that males actually seem to be fooling the dominant males because they also receive a lot of courtship. So they receive similar levels of courtship to females. And so we think that the spot during this particular life stage is effectively fooling the dominant males into not beating up the juvenile males. And that this could have a survival advantage. So then why is it polymorphic in all these populations if it's contributing to survival? Well, females are not fooled. Even when males go on to develop their other secondary sexual characteristics, females have a clear preference for males without the spot. And so they, so we're kind of envisioning this tug of war between the female preference and the male survival in maintaining the polymorphism in this trait. Okay, so these studies hint at behavioral consequences of the trait, why it might be polymorphic in so many species. We next tackled its genetic basis. Again, a shout out to pigmentation traits where we keep getting GWAS peaks like this. Tristad, who led this project, did a GWAS of males with and without the false gravid spot. He sees this really nice peak. This peak actually completely localizes to an intergenic region upstream of this gene known as KIT ligand, which is a really important pigmentation gene in mammals. What I was just like thrilled about this, this association with the pigmentation gene, but Tris was really bothered by the shape of this peak. And he convinced me to do a lot of long-read sequencing, which I'm about to tell you about. And at the time, I thought it was maybe a little excessive, but I have completely changed my perspective. To give you some background on the plots I'm about to show you, I'm going to be showing you alignments of these long-read assemblies of males with different phenotypes. If you see something like this, that means that the assemblies are completely collinear. That doesn't mean there aren't SNP differences. It just means there aren't structural differences between them. If you see something like this with this sort of cross pattern, that's an inversion. And if you see these overlapping assemblies, those are duplications. Okay, on the next slide, I'm going to show you an alignment of a false gravid haplotype, a spotted haplotype, to a wild type. What we see is this really complex structural variant, but it's not very big. It's only about 30 kb in size, and it precisely localizes with our genome-wide association signal. And so we believe that this variant is what's actually driving the signal in this region. It also happens to be just upstream of the KIT ligand A promoter. And so this sort of led to the hypothesis that this variant is driving some change in expression that then generates the trait. But before we sort of move on to that hypothesis, Tris really wanted to be sure that it was this structural variant that was associated with the trait. And so he did a bunch of long-read assemblies and was able to show that consistently individuals with the rearrangement, individuals with the spot have this complex rearrangement. And so it's not just a one-off, it's always associated with the trait. And all wild-type individuals have this sort of non-rearranged haplotype. Okay, so getting back to the hypothesis about how this trait is generated, the idea is that this rearrangement causes some regulatory difference in driving the expression of this allele. When KIT ligand A is upregulated, it causes migration of melanocytes to the region of expression. And so this would provide a really intuitive explanation of the trait. He started with just some bulk RNA-seq, looking at differences in expression in this particular tissue between males and with and without the spot. He sees really clear upregulation of KIT ligand A, as well as other melanocyte-related genes. But what is quite striking is if he looks at just the tissue above, the muscle that's above where those melanocytes accumulate, he sees no difference in expression. And so this is suggestive that it's actually something very specific about this tissue environment and the structural rearrangement that's driving the expression difference. So to try to test this explicitly, we generated crosses between individuals with and without the rearrangement, and then track the allelic expression. And we see exactly what you would expect, where individuals with the rearranged haplotype have high expression of that allele only in this tissue. Whereas in other tissues, we see this one-to-one expression of the two alleles. Okay, so this provides sort of a clear explanation of the development and patterning of the trait. Now we can turn to the exciting questions, which are studying the evolutionary history. Okay, so I mentioned earlier that this trait is polymorphic, but that it's also oddly distributed. So if we look at this phylogeny, the purple are the individuals with the trait, it seems kind of clear, like if we look up here, it doesn't look that confusing. If you look at the platyfish, it seems clear that the common ancestor probably had the trait, it was lost once, no big deal. But if we look down here, it's really unclear what's going on. So we have these sort of places where the trait is popping up, and it's not really clear what was likely going on in the common ancestor. The first thing Tris did is map the trait in a representative of all the major groups. And this is what his results look like. It was a combination of QTL mapping and GWAS. And he basically sees, this clear signal that it's always KIT ligand A in all of these species. So it's not the case that, different genes are being involved and generating the same trait. Okay, so finding that it had the same genetic basis, we next wondered whether it might be passing between species due to hybridization, or potentially independently evolved via the same gene. And so to distinguish between that, we have focused in on this group that we study a lot in lab for our other work, and also capture some of this representation of these weird cases where the trait's popping up where we might think it shouldn't in the phylogeny. So this is a group that we've studied intensively in our natural collections. It's a little hard to see here with the zoom, but where we have lots of natural populations that we sample. These species, Cortezi and Birchmai, both have the spot, and Malinche does not. But Cortezi and Birchmai co-occur with another species that's distantly related, Xiphophorus variatus, in which the spot is very common. And so we began to start considering the possibility that even though these guys are about 6 million generations diverged from Birchmai and Cortezi, that they might have rare hybridization events that could have resulted in the spot transfer. And so this became a more compelling hypothesis due to other work in the lab where we actually detected natural hybrids between variatus and Cortezi and between variatus and Birchmai. So it seems clear that hybridization between these groups can occur. And now we wanted to ask, did it result in the movement of this allele? Okay, so of course, Tris really wanted to do a long read assembly of this individual of variatus. And at this point, I was convinced, so we went ahead and did it. What he finds is the sort of rearrangement haplotype that's present in variatus is very similar to what we see in Birchmai. So that's sort of like a qualitative look. To look at it quantitatively, he collected all the long read assemblies that we had generated and that our collaborators had generated, and aligned this region. We only use long read assemblies because the short reads in this region are a mess. And then use this alignment to infer phylogenetic relationships. Okay, so this is what he sees. So if we look first at the wild type birchmani individuals, ones that don't have the spot, they all cluster down here with their close relatives, Malinche and Cortezi. But if you look at the spotted birchmani, those haplotypes are embedded within this deeply divergent platyfish group and most closely related to this variatus species that co-occurs with it. So this is quite suggestive of directional movement of the spotted haplotype from variatus into birchmani. Okay, so here we have sort of compelling evidence that hybridization explains the appearance of this trait in birchmani. But what about these other cases? So birchmani has long been known to hybridize with several other species. One of these species is its sister species, Malinche. This species does not normally have spots, but in collecting in natural populations, we found one population in a place called Teripancalcho where Malinche individuals were spotted. And so Tris went ahead and sequenced these individuals and inferred the phylogenetic relationship. So looking here at these individuals that are abbreviated Teddy, we see that they are embedded phylogenetically with the birchmani individuals that are spotted, and in turn with all of these platyfish. So they're basically nesting into that group of divergent haplotypes, whereas the rest of the Malinche individuals that are not spotted are with their close relatives. Okay, we took this a little further and used local ancestry inference to look at the ancestry assignment in these spotted individuals versus unspotted Malinche, and see evidence of introgression of a haplotype that's actually quite large from birchmani, suggesting a recent event. And so overall, this suggests a really complex evolutionary history of this trait, where it was introduced to birchmani via hybridization with variatus, and then variatus and Malinche do not have contact zones. And so it must have passed through birchmani into Malinche. And so we have this sort of conduit event where we're passing these haplotypes through different species. Okay, so just to kind of wrap up here, this isn't the whole story. It's not all hybridization mediated, we think. Because you could imagine that this other species, Cortezi, which is also spotted, could have the same thing going on. This species also hybridizes with birchmani. You could have the same mechanism passing that into Cortezi, or even directly introducing it from variatus. And we know that the spotting in Cortezi is explained by the same gene. But when we look at the haplotype in Cortezi, it's structurally quite different. And if we look at the phylogenetic placement of Cortezi, it is not nested with the platyfish. It's closely related to other individuals in its group. And so we haven't quite figured out what's going on with this individual, but we think there's a possibility of recurrent evolution, at least in some cases. And I think there's a lot more to delve into here as we explore the rest of the group. So just to wrap up, we think that there's, in this particular trait, cases of both de novo and repeated evolution through introgression of this same regulatory region. Okay, so I'm going to conclude here by saying today I told you about two really different cases where we can track, we can understand the genetic mechanisms underlying trait variation. We can track its evolution that's led us to really different evolutionary processes in terms of recent de novo evolution of a trait versus sort of remixing of this older variation via hybridization. And I think The strategy of focusing on these genetically simple traits allows us to get a lot deeper in terms of disentangling the mechanisms, but of course it's not the whole story. We know that many traits are polygenic, and it's going to be a lot tougher to understand their evolutionary paths. Okay, to conclude and zoom back out, our lab group is most excited about asking questions about how organisms adapt to their environments and form new species by connecting genetics and molecular biology to evolutionary biology. And we think this will help us understand trait variation in nature. And by thanking my lab and collaborators and funding, happy to take any questions.
Yeah, great talk. It seemed like you were like very careful to like avoid calling any of these mechanisms like being under selection. And I think that's probably warranted given how hard it is to test non-traditional mutations for selection. But it was kind of, yeah, so I kind of wanted to know on how you might be like trying to directly actually measure selection on these like complex genome mutations.
Yeah, so a lot of the sort of traditional tests for selective sweeps do not work. Well, for example, like if you run them on the transposable element insertion, it's like a screaming signal because it's so young, and they're like not designed to test that kind of mutation. So it's like a large region that has no diversity that looks like a signature of selection, but I think we don't really know. In the second case, with the false gravid spot, we think there's very compelling evidence of balancing selection. So, it's a trait that's found as a polymorphism across, almost a dozen species. It's maintained as a polymorphism. We can see trade-offs. We've actually done similar behavioral tests in other species, and it seems to be this aggression mate preference trade-off in general. So that I would feel pretty confident saying it's under balancing selection. I think with the iridescence, it's quite complicated because it's so recent. And we can see these functional effects of the trait. It seems kind of, it's such an obvious trait, like, but it's been quite hard to rule out neutrality. So yeah, kind of holding that possibility. So.
Yeah, the second case seems more, you said tug of war, and my tug of war
Yeah.
is classic balancing
Yeah.
selection. But yeah, and I guess my other question was like, in the first case, you have like a mechanism and impact. In the second case, it sounds like you're still like looking for the mechanism that's causing the
Oh, yeah. So the rearrangement. So yeah, this has been very interesting thing, and I spoke with several people about this today. So the rearrangement is not transposable element mediated. However, it occurs in these sort of like hotspot of non-beta DNA. So it might be predicted to be fragile. However, the best evidence we have about what's going on is from the multiple individuals we've sequenced, because we actually see I'm gonna maybe mess up the numbers, but we have a total of 18 haplotypes with the rearrangement, and 16 of them are different from each other in terms of some element of the structure. So usually it's the number of repeats, the inversion is always there, or sort of the length of some of the elements. So to me, that suggests high mutability, but we have not directly tested that.
Question about, I guess, the sparkliness of the birds. Do you measure, is that just measured with like a camera measuring intensity of the reflection or something like that?
It's with an optic fiber, but I know that's about as much as I know about the methodology. Yeah.
Like they're looking at, because I think birds like to see
In CNUV, yeah. I think Paula tested into the UV, and fish can generally see in the UV as well. Let me pull up that result if I can find it. Yeah, so that's the range she tested, so mainly visible. I don't know how birds see UV through water, and that's another element here.
Like, couple steps.
Yeah, yeah. So, yeah.
For the sparkle phenotype, you said that at least every individual has at least one copy of this deletion. Do you see differences in like the expression, or are there biases in like males and females, or
Yeah.
that between homozygous and heterozygous for deletion?
So we do think that the homozygotes have a higher intensity of the trait, and the data is a bit noisy, but they do also tend to have higher ALK expression. There's no difference in the trait between males and females, and we've done mate preference trials, and we see no preference difference between. We've done it in females for like sparkle versus non-sparkle males.
For the second case, I guess the two quick questions. The first is, have you tried to like date the age of the inversion?
So the inversion itself, we've tried to date the age of the introgression, but we haven't done anything that I think would date the age of the inversion itself. We might need a few more species. We have, let's see. So these are the species that we have well sampled, and they all have the inversion. This one actually has a deletion in this region. And so that's also kind of a nice mechanistic piece. So you look at the alignment, it's just gone right in this region where the inversion is. These guys we haven't looked at, so we don't know exactly the age of the inversion. This clade is about like 2 million years old to 4 million generations. The introgression is not super recent. So it's more, so the divergence between Birchmine variatus, like at the nuclear level, is almost 2%. And if you look in this inversion, the divergence between them is a little less than 1%, but much higher than you would expect for like within population variation.
And so for when you're calling these introgress regions, given like how the place of this gene flow seems to be, how are you, I guess, like deciding who are going to be the source species? Because I can imagine, right, that if you paint it a bunch of different ways, you could do different color paintings to, yeah, I guess try to piece out where it might have this originally.
You mean like the source species up here? So this is somewhat of an assumption, but variatus is the only one that overlaps with any of these species. It's a widespread species, so nobody else is in the area. Now, it's possible that there could have been some ghost lineage that had the same, it's old, the introgression appears old enough that, there could have been a species that went extinct that also lived in this region, but probably nothing, in terms of like these other extant species contributing the haplotype. Thanks.